Hello, this video series will capture docking with emphasis on docking validation. The process will apply to general docking for ligand receptor interactions. The series will also entail how to set a flexible residue in the receptor, how to generate the docking parameter file, grid file, and the docking proper. And finally, how to analyze the docking results in the DLG. First, we are going to use Autodock and we need to set our working directory. This can be done by simply using the set option in the menu there. And you can copy your working directory and you paste here and set. So since it's startup directory, sometimes you may need to restart Autodock for this to take effect, but I have already done mine. Next, we are loading the protein molecule. In this case, working with 1GSF, I made a copy because I'll need that for the co-crystallized ligand later, since we are discussing validation. So I'll select that. Now we can see that our enzyme is up. It has 4 monomers, and we can look further to see if there are some other things that are here. Specifically, you can see we have water, 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 water. All of these we need to take off before we start. And this is the co-crystallized ligand. So from edit, we can just delete all the water in the entire protein molecule. Once that is done, we can select chain B, chain C, and chain D. Oh, okay, let's delete them one after the other. Normally I delete them together, but it's not responding now. Okay, come to delete selected atoms, continue. Then we have this, go back again, delete. Following the same process, we have deleted all the other chains, and we can now take a look at chain A, which we are using for this training. And we have the standard amino acids, and here we have itacrinic acid. So if we bring this one further, better view, trying to see, if we select, we can see that's itacrinic acid there. That's the non-amino acid ligand. Since we are cleaning to get the protein ready for docking, we will come to the same function, delete it, continue, and you can see it's gone. To repair missing atoms, we can now come again to edit in the menu bar, miscellaneous, repair missing atom, and then you give this one time to run. It may take a few minutes. It's important to do this because if the atoms are not repaired, you may have issues with the docking later. It took over 3 minutes for the repair to be completed. Once done, just click save as 2 sets and then dismiss. Okay. Then come back, hydrogens, add, polar only. Once that's done, back to edit again, charges, this time add command charges, total added. Okay. You can always confirm by going back again to see how many, check totals on residues. So once you see this, it means all the charges have been added. Back again to edit, This time you come to atom, assign AD4 types. This is important too. So that's done. What we have done so far is clean our receptor and it's now ready for use.